Hello everyone, very good evening. In today's lecture, we will be talking about composition of DNA. So, let us see very basic things about DNA. DNA is a polymer of nucleotide. That means many nucleotides are attached together to synthesize DNA. It is said that master plan of life is present in DNA. Master plan of life is present in DNA. The full form of DNA is deoxyribonucleic acid. Deoxyribonucleic acid. Now, as DNA is polymer of nucleotide, let us see what are nucleotides. So, nucleotides consist of three components, that is pentose sugar, second one is nitrogenous bases, and third one is phosphate group. Now this pentose sugar and nitrogenous bases are together called as nucleosides. And when you add phosphate group to those nucleosides, you will get nucleotide. So, DNA is a polymer of nucleotide, and nucleotides consist of pentose sugar, nitrogenous bases, and phosphate group. Pentose sugar and nitrogenous bases are together called as nucleosides. And when you add phosphate group to nucleosides, you will get nucleotides. Okay, I hope you have got this basic structure of DNA. Now let us see one by one what are pentose sugars, nitrogenous bases, and phosphate group. So first we will talk about pentose sugars. So from the name itself, you will get that pentose. Pentose, that means any 5 carbon compound. So any 5 carbon sugars are called as pentose sugars. So usually there are two types of sugars. First one is ribose sugar, which is usually present in RNA, and second one is deoxyribose sugar, which is present in DNA. So let us see the structure of these pentose sugars. Now see, Now, this is the structure of ribose sugar. So, this is carbon number 1. This is carbon number 2. This is carbon number 3. This is carbon number 4, and this carbon is carbon number 5. These sugars or these carbons are named as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, because we are giving numbering to our nitrogenous bases as well. So to become easy, whether we are talking about sugars or we are talking about nitrogenous bases, we are giving numbering to carbons of ribose sugar as 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. Now, during change in pH, sometimes this hydrogen atom is removed at hydrogen atom which is present at 2 prime carbon is removed, and this oxygen acts as nucleophile, and this oxygen then will attack phosphodiester bond and it will hamper the structure. So RNA is not stable, and making this unstable group as a genetic material is not advisable. So nature chose DNA as a genetic material. So in DNA, in the pentose sugar of DNA, this oxygen atom is removed. So the structure of pentose sugar in RNA is as follows. So the only difference between structure of pentose sugars in RNA and DNA is the oxygen atom which is present at 2 prime carbon is absent. Thus it is called as deoxyribose sugar. This is the only difference between the pentose sugars of RNA and DNA. So what we have seen that in RNA at 2 prime end OH group, so not at end, at 2 prime carbon hydroxyl group is present, but during change in pH hydrogen atom is sometimes removed at 2 prime end, and thus oxygen acts as a nucleophile, and this nucleophilic oxygen attacks phosphodiester bond. Thus RNA is not stable, and making this unstable group as a genetic material is not advisable. Thus nature chose DNA as a genetic material as this hydroxyl group is absent at 2 prime OH. In DNA, oxygen is removed and only hydrogen atom is present at 2 prime end. Now we will see or we will talk about nitrogenous bases which are present in DNA. Now we are talking about nitrogenous bases. So there are two types of nitrogenous bases. There are purines and pyrimidines. Purines consist of 2 rings and pyrimidines consist of 1 ring. So in purine, 1 ring is of 6 carbon and second ring is of 5 carbon, and pyrimidines consist of only 6 carbon ring. Now purines are adenine, guanine, and pyrimidines are thymine, or this thymine is present in DNA, and instead of thymine, uracil is present in RNA. And third one is cytosine. The difference between thymine and uracil is only the presence of methyl group in thymine. Now let us see the structure of adenine, guanine, thymine, uracil and cytosine one by one. So first we will talk about adenine, purines. So first we will talk about purines. So purines consist of two rings. Okay. Now the basic structure of purine. So first we will see the general structure of purines. So in purines, this is your six carbon ring and this is your 5 carbon ring, and you will number it as 1 2 3 4 5 6 7 8 9. So the 6 carbon ring in purines is clockwise and 5 carbon ring is numbered anti-clockwise. Okay. Now at carbon number 1 and at carbon number 3 of 6 carbon ring, nitrogen atom is present, and at carbon number 7 and carbon number 9 of this 5 carbon ring, nitrogen atoms are present. This structure is exactly same in case of guanine. This is the basic structure of purine. We will also see how you can remember the structure of nitrogenous bases. That's why I'm telling you all these things. It is 1 2 3 4 5 6 7 8 and 9. Again, as I said, at carbon number 1, carbon number 3 of 6 carbon ring, and at carbon number 7, at number 7, not carbon number, I'm sorry, at number 7 and at number 9 you have nitrogen atom. Now the basic difference between adenine and purine. So this adenine, which is also called as 6-amino purine, and guanine is called as 2-amino 6-oxo purine. So from this what you will get? 6 amino. That means at carbon number 6 you will get amino group, and in case of guanine at carbon number 2 there is amino group, and at carbon number 2 is amino group, and at carbon number 6 there is oxo group. This is how you will draw the structure of nitrogenous bases, that is purine, which are adenine and guanine. And now I am explaining it to you again. Now see, the basic structure of purine, it consists of two rings, that is one ring is of 6 carbon and another ring is of 5 carbon. The numbering of 6 carbon ring is always clockwise in case of purine, and 5 carbon ring is always numbered anti-clockwise. At carbon number 6 in case of adenine there is amino group, while in case of guanine there is oxo group. At number 1, 3, 7 and 9 of purine there are nitrogen atoms. This is how you will remember the basic structure of purine. And then at carbon number 6 of adenine you will draw amino group, and at carbon number 2 of guanine you will draw amino group, and at carbon number 6 of guanine you will draw oxo group. This is the basic difference in adenine and guanine, which are purines. Okay, now we will talk about pyrimidines. We will talk about pyrimidines. Pyrimidines consist of only one ring, one 6 carbon ring. So it will become easy for you to remember pyrimidine. So pyrimidines are thymine, which in case of RNA is uracil, and third one is cytosine. Thymine is present in DNA, uracil is present in RNA. Now again we will draw 6 carbon ring in again we will draw 6 carbon ring for pyrimidines. Now these pyrimidines are numbered anti-clockwise. So this is 1, 2, 3, 4, 5, 6. In purines, the 6 carbon ring was numbered clockwise, but in pyrimidines it is numbered anti-clockwise. Please remember this. Again 1, 2, 3, 4, 5 and 6. 1, 2, 3, 4, 5, 6. Now in case of purines, at number 1 and at number 3 you have nitrogen atoms in all cases. Okay. So in pyrimidines, thymine contains oxo group. So this is oxo group. Uracil contains oxo group at 4th position, while the cytosine at 4th position contains amino group. Now the basic difference between uracil and thymine is, in DNA, thymine at 5th position contains methyl group, while methyl group is absent in uracil. So it is said that uracil in DNA has CH3 at fifth carbon. Uracil in DNA has CH3 as fifth carbon, which is named as thymine. So I hope you have got this. What we are saying, pyrimidines is a 6 carbon. Pyrimidines consist of 6 carbon ring, and they are thymine, uracil, cytosine. 6 carbon rings are numbered anti-clockwise in case of pyrimidines. At number 1 and 3 there are presence of nitrogen atoms in all pyrimidines, and the basic difference is cytosine has amino group at 4th position, whereas thymine and uracil has oxo group at 4th position. And the basic difference between thymine and uracil is at 5th position. In case of thymine, there is presence of methyl group. Hope you have got this point. Now again I will complete this structure by drawing the bonds over here. Okay. Now, how these ribose sugars, or you can say pentose sugars, and nitrogenous bases are joined together. These nitrogenous bases and pentose sugars are joined together by N-glycosidic bond. They make N-glycosidic bond. Between pentose sugars and nitrogenous bases. Now, in case of purines, nitrogen present at 9 number makes glycosidic bond with 1 prime carbon of pentose sugar. So in purine it is nitrogen at 9 position will pair up with carbon at first position present in sugar. This is in NB and this is in pentose sugar. Or you can say ribose sugar. Whereas in case of pyrimidines, nitrogen present at first position will make glycosidic bond with first carbon of pentose sugar. So it is 1-1. While in case of purine it is 9-1. Now let us see how they make bond in case of purines and pyrimidines in short. So this is your ribose sugar. This is 1, this is 1 prime, 2 prime, 3 prime, 4 prime, and it is 5 prime. So at this position purines, that is nitrogen present at position number 9 of purine will make bond, while and this bond is N-glycosidic bond. While in case of pyrimidines, again this is your ribose sugar and nitrogen, which is present at first position of pyrimidine, will make bond with, or you can say will make N-glycosidic bond with ribose sugar. Please ignore the sound. Sorry for the disturbance. Now next we will talk about the phosphate group, which is the last part of nucleotides. Third one is phosphate group. This is your phosphate group. Okay, now as we have seen the basic structure of nucleotides, let us see how the polymer is synthesized, or how one nucleotide is attached to second nucleotide with the help of phosphodiester bond, or we will see how phosphodiester bond is synthesized. Now see, this is your basic structure of nucleotide, or you can say this is the pre-existing nucleotide. We will give name phosphodiester bond synthesis. So so this is your ribose sugar, which is attached with the nitrogen base, and this is phosphate group. This is base. Now this is a pre-existing nucleotide which is present in environment. and this is an upcoming nucleotide which is coming in the chain. Now this 3' OH group which is present in the pentose sugar will attack this phosphate of upcoming nucleotide. This is upcoming nucleotide. The 3' OH group which is present in pre-existing nucleotide will attack 5' phosphate group of upcoming nucleotide. This phosphate group is present at 5' end, and thus now during this reaction what will happen, the hydrogen atom will be taken up from upcoming environment. During bond formation this hydrogen will be lost, and again this oxygen will be lost, and there will be synthesis of H2. So 3' OH of pre-existing nucleotide will attack 5' phosphate group of upcoming nucleotide, and there will be formation of bond. So this was your pre-existing nucleotide, and this 3' OH, when it will act the 5' end of 5' phosphate, it will form bond with 5' phosphate. This oxygen atom is as it is. The hydrogen atom will pair up with this H plus ion which is already present in environment. Again this phosphate group. Now this is your upcoming nucleotide. This is please. Okay, so during this reaction H2O is released, and this is how 3' OH of pre-existing nucleotide will attack upcoming nucleotide, and there will be formation of phosphodiester bond. Phospho diester bond formation. So if you want to write, you can write three prime OH of the pre-existing nucleotide attacks 5 prime phosphate of the upcoming nucleotide. And in DNA, bond formation, I am talking about bond formation, is from 3 prime end to 5 prime end. This 3 prime end is attacking 5 prime end. That is why bond formation in case of DNA is from 3 prime end to 5 prime end. Now let us see one rule, that is Chargaff's rule, or you can say it is a just a minute. Let us talk about Chargaff's rule. In this rule, what the scientist did, he did paper chromatography, and he observed that the nucleotides which are present in DNA or RNA are not present in equal amount. So from his experiment, he found out few observations, and there were 4 different nucleotides are not present in equal amount. That is, their exact ratio varies from one another. See, as adenine pairs with thymine, if you have 5 adenine, number of thymine will be 5. Adenine is purine, thymine is pyrimidine. Again, cytosine pairs with guanine. This is guanine is purine. Cytosine is pyrimidine. If you have 5 cytosine, you have 5 guanine. So you will say that number of purines equals to number of pyrimidine. That is, if you have 5A, you will have 5T. If you have 5G, you will have 5C. That means number of purine is same as number of pyrimidine. But if you will see, number of AT and number of GC is not same. Sometimes number of AT are higher in case of humans because there are repetitive sequence in humans. But in case of bacteria, GC content is higher. So AT and GC ratio is not same. It varies in almost all species. In some case it could be equal, but it is very rare. It is usually different in different species. So you can write in case of bacteria, AT content is less and GC content is higher. And in case of humans, AT content is higher as there is repetitive sequence and GC content is lesser. So this is all about composition of DNA. I hope you have got the lecture. If you have any doubt, you can comment me in the comment section. Thank you so much and have a nice day and keep studying. Bye bye.